Hello students, welcome to Read MedPrep Academy channel. Today in Molecular Genetics Part 1, we are going to introduce you to the molecular genetics. We are going to discuss about the gene and the genetic material. Mendel's theory dispelled the mystery of why traits or characters seem to appear and disappear magically from one generation to the next. Mendel's work reveals the patterns of heredity and reflect the transmission of evolved information from the parents to the offspring. This information is located on the chromosomes. Each chromosome carries the genetic information in the form of genes, and the arms of the chromosome are called chromatids that divide longwise during cell division. The centromere is the place where the chromatids get connected. One of the most advanced realizations of human knowledge was that our unique characteristics are encoded within the molecules of DNA, deoxyribonucleic acid. The discovery that DNA is the genetic material left several questions unanswered. How is the information in the DNA used? Scientists now know that DNA directs the construction of the proteins. You can see that a gene can code for a particular protein. Another gene can code for another protein. So proteins determine the shapes of the cells, the rate of the chemical reactions, such as those that occur during metabolism and photosynthesis. What do proteins do? Proteins perform a wide range of biological functions. As enzymes, they catalyze the various reactions. Carrier proteins transport the molecules across the membranes. Antibodies defend against the diseases. Structural proteins support the cells and the tissues. Hormones transmit the information. Transport proteins such as hemoglobin carry oxygen. Contractile proteins enable the muscles to contract. What is the genome? The hereditary nature of every living organism is defined by its genome, which consists of a long sequence of nucleic acids that provide the information needed to construct the organism. A genome is the full set of genetic information that an organism carries in its DNA. It is otherwise called the genetic constitution of an individual. The study of any genome starts with the analysis of the chromosomes. Chromosomes are nothing but bundles of DNA and proteins found in the nuclei of the eukaryotic cells. What does the genome contain? The genome contains the complete set of hereditary information of an organism. What is genomics? Study of an organism's entire genome is called genomics. All the DNA that is encoded in the organism, the nucleus, the mitochondria, the chloroplast, and the genes and the stuff in between them. What made genomics possible? High throughput sequencing technology, availability of many landmarks, bioinformatic techniques to assemble sequences. Where is the gene? The genome may be divided into a number of different nucleic acid molecules. Each of the nucleic acid molecule may contain large number of genes. Each gene is a sequence within the nucleic acid that represents a single particular protein. Here you can see the cells contain the nucleus. The nucleus contains the chromosomes. The chromosomes contain the deoxyribonucleic acid. And the deoxyribonucleic acid contains the number of genes. Each gene codes for a particular protein. And the proteins are responsible for the structure and function of the human body as well as the organism. What is the functional unit of inheritance? A gene is a basic physical and functional unit of heredity. The concept of gene was first explained by Gregor Mendel in 1860. Mendel never used the term gene. He called it as a factor. In 1909, the Danish biologist Wilhelm Johansen coined the term gene that was referred to discrete determinants of the inherited characteristics. What are genes? According to the classical concept of gene introduced by Sutton in 1902, genes have been defined as discrete particles that follow the Mendelian rules of inheritance. They occupy a definite locus in the chromosome and they are responsible for the expression of a specific phenotypic character. The number of genes in each organism outnumbers the number of chromosomes. Hence, several genes are located on the same chromosome. What is called a locus? The genes are arranged in a single linear order like the beads on a string. Each gene occupies a specific position called locus. Here you can see that a single chromosome which has a centromere and the upper arm and the lower arm having multiple genes. And the sister chromatids have similar genes in the sister chromatids. And during the meiotic division, chromosomes replicate during interphase before prophase 1 and they form a pair of homologous chromosomes. The genes are located at a specific loci. For example, you can see here that a gene located in the upper arm codes for the eye color, which is brown. Similarly, a gene in the lower arm of the chromatid codes for a hair color genome, hazel color of the hair. What are alleles? Genes exist in several alternate forms called alleles. Genes are capable of self-duplication, producing their own copies. Here you can see that the alleles may be homozygous for the dominant allele represented by the capital letters. The alleles may be homozygous for the recessive allele. They are represented by the small case letters, or sometimes they may be heterozygous. Heterozygous, dominant and a recessive allele. Only the dominant character will be expressed in a heterozygous. What is mutation? Genes may undergo sudden change in positions and compositions in the chromosomes. They are called mutations. Here you can see a normal sequence GAG, guanine, adenine and guanine. This normal sequence is producing a normal fertilization embryo on a normal phenotype, whereas a mutated gene where the adenine is replaced by thymine. This causes an altered phenotype because of the alteration in the gene sequence. One gene, one enzyme hypothesis. Experiments of George Beadle and Edward Tatum in early 1940s on the Neurospora crassia, the red bread mold, led them to propose one gene, one enzyme hypothesis, which states that one gene controls the production of one enzyme. Here you can see in 1942, the hypothesis was proposed where bread mold Neurospora can normally grow on the minimal media because it can synthesize most essential metabolites. In this biosynthesis is under genetic control, then mutants in these genes would require additional metabolites in their media. This was tested by irradiating Neurospora spores and screening the cells they produced for additional nutritional requirements. Auxotroph. Wild type spores of Neurospora were irradiated with X-rays and ultraviolet rays, and the resulting strains were crossed with the wild type, and they were grown in a medium, and the medium showed that the vitamins were very much essential for the growth of these Neurospora. One gene, one polypeptide hypothesis. It was observed that an enzyme may be composed of more than one polypeptide chain, and the gene can code for only one polypeptide chain. One gene, one polypeptide hypothesis indicates that the DNA contains all of our hereditary information. Genes are located in our DNA. 25,000 genes in our DNA in 46 chromosomes. Each gene codes for a specific polypeptide. You can see here that specific gene codes for a specific enzyme, and each enzyme codes for a particular amino acid. Likewise, gene 2 you can see it codes for citrulline, and gene 3 you can see it codes for arginosuccinate, and enzyme H codes for arginine. So the one gene, one polypeptide evidence shows that each gene is responsible for production of one polypeptide. Thus, the hypothesis states that one gene controls the production of one polypeptide chain of an enzyme molecule. Now let us move on to the search of genetic material. The discovery of chromosomes as early as 1848, Wilhelm Hofmeister, a German botanist, had observed that cell nuclei organize themselves into small rod-like bodies during mitosis called chromosomes. You can see here that the nucleus gets organized into rod-like bodies during the mitosis, and these rod-like bodies called chromosomes separate by the microtubules during mitosis, forming two daughter cells. In 1869, Friedrich Miescher, a Swiss physician, isolated a substance from the cell nuclei and called it as nuclein. Nuclein was renamed as nucleic acid in 1889 and is now known as deoxyribonucleic acid. Richard Altman found that nuclein was an acid, hence the name he gave was nucleic acid. In 1919, Phoebus Levine identified the components of the nucleic acid. What is present in the chromosomes? By 1920, it became clear that chromosomes are made up of proteins and deoxyribonucleic acid. Many experiments were carried out to study the actual carriers of genetic information. And the chromosomes are basically composed of proteins and a single molecule of DNA. What is the genetic material? Griffith's experiment proved that DNA is the genetic material. Bacterial transformation experiments provided the first proof that DNA is the genetic material. Here you can see the hypothesis that material in a dead bacterial cells can transform living bacterial cells. How was this experiment done? A living smooth strain which was found to be virulent was injected into a mouse, and the mouse dies having the S strain. Then a living R strain, a non-virulent strain, R means rough strain, was injected into a mouse. The mouse lives and there is no S strain inside the mouse, and in another set the S strain, which is the smooth strain which is virulent, is heated and deactivated and is made avirulent and is injected into the mouse, and the mouse lives, and there is no living S strain in the mouse. Then in the last experiment you can see that the living rough strain and the dead smooth strain were mixed and were injected into the mouse, and the mouse dies. On examination it was found that it was having the smooth strain which was virulent. So finally it was concluded that a chemical substance was transferred from one cell is genetically to the another cell, that is from the S strain to the R strain. Griffith's experiments in 1928 first demonstrated that genetic information could be transferred without knowing that this information was, he called it the transforming principle. It was the first step towards proving that nucleic acids and not the proteins are the chemical basis of experiment. Griffith could not understand the cause of bacterial transformation, and the biochemical nature of genetic material was not defined from his experiments. Later, Oswald Avery, Colin MacLeod, and Maclean McCarthy in 1944 repeated the Griffith's experiments in an in vitro system in order to identify the nature of the transforming substance or the principle responsible for converting a non-virulent strain into a virulent strain. Here you can see Oswald Avery, Maclean McCarthy, and Colin MacLeod, who repeated the Griffith's experiments. What they did, they took a rough strain, R strain, which was non-virulent, and injected into the mouse, and the mouse lives. Then they took a smooth strain which was virulent and they injected into the mouse. The mouse dies, indicating that the smooth strain is virulent. Then they kill the S strain or the smooth strain and they injected into the mouse. Then the mouse lives. This shows that the S strain was made avirulent because of heat. Then the rough strain, which is non-virulent, and the smooth strain, which was heat killed, made A virulent was injected into the mouse and the mouse dies. This showed that the transforming principle was transformed from the S strain. This indicates that there is some transforming factor that is transferred from one cell to another. They observed that DNA, RNA and proteins isolated from the heat-killed S strain when added to the R strain changed their surface character from rough to smooth and also made them pathogenic. They also did experiments with the enzymes, DNAs, RNAs and proteases. How they did? When the extract was treated with DNAs, an enzyme which destroys DNA, the transforming ability was lost. Similarly, they took another sample and they added RNAs, an enzyme which destroys RNA, and proteases which destroys the proteins. This showed that the transformation principle was not affected. According to this hypothesis, the genetic material of the cell is either protein or nucleic acid, DNA or RNA. So in this experiment, you can see that the sample is taken and the lipase and sugars are removed and heat-killed S cells or smooth cells are added. In one sample, proteinase is added and it is devoid of protein now. It contains DNA and RNA. And R cells are added to this sample and it showed that the S cells appear. In another sample, only RNase is added. That means RNA is removed and it contains the protein and DNA. And to this sample, R cells are added. This showed that the S cells appear. In the third sample, the DNase is added. So the sample contains the protein as well as RNA and DNA is removed. And to this R cells are added. This shows that the S cells did not appear in this sample. So removal of lipids and sugars from a solution of heat-killed S cells, proteins, RNA and DNA remain. These solutions are treated with various enzymes to remove the protein, RNA or DNA and added to this culture containing living R cells. They are observed for transformation by testing the presence of virulent S cells. According to the conclusion of this experiment, transformation requires DNA. Therefore, it is the genetic material of the cell. Here you can see in this slide S strain extract which was devoid of polysaccharides when given. Since it contained DNA, the mouse dies. The second sample in which the lipid is removed, it contains proteins, RNA, DNA, the mouse dies. In the third sample which is removed of RNA, again the mouse dies since the DNA is present. In the fourth sample where the protein is destroyed, the DNA and RNA are present, the mouse dies. In the last sample where only the DNA is destroyed, it contains protein and the RNA, the mouse lives, indicating that DNA is the genetic material and no live S strain have been recovered. What is the conclusion of this experiment? This experiment suggests that DNA and not the proteins is the genetic material. The phenomenon by which the DNA related from one type of cell, S strain, when introduced into another type of the strain or R strain, is able to retain some of the properties of the S strain, and this is referred as the transformation. Many biologists, despite their early experiments of Griffith, Avery and others, still believe that protein and not the DNA was the hereditary material in a cell. As eukaryotic chromosome consists of roughly equal amounts of protein and DNA, it was said that only the protein had sufficient chemical diversity and complexity to encode the information required for genetic material. In 1952, however, the results of the Hershey-Chase experiment finally provided the convincing evidence that DNA is the only genetic material. According to the Hershey-Chase experiment, they used T2 bacteriophage. Alfred Hershey and Maratha Chase in 1952 conducted experiments on the bacteriophages that infect the bacteria. Phage T2 is a virus that infects the bacterium E. coli. When phages or the viruses that infect the bacteria are added to the bacteria, they adsorb to the outer surface. Some materials enter into the bacterium, and later each bacterium lyses to release a large number of progeny phase. Here you can see that any virus that infects a cell gets incorporated into the bacterial DNA or the cell DNA, or it uses the cell DNA and multiplies itself and causes lysis of the cell, and large number of phages are released. Hershey and Chase wanted to observe whether it was DNA or protein that entered the bacteria. All nucleic acids contain phosphorus and proteins contain sulfur in the amino acid cysteine and methionine. Experimental use of radioisotopes. Hershey and Chase designed an experiment using radioactive isotopes like sulfur 35 and phosphorus 32 to keep separate track of the viral protein and nucleic acids during the infection process. So the phages were allowed to infect the bacteria in a culture medium which containing the radioactive isotopes S35 or P32. So the bacteriophage that grew in the presence of S35 had labeled proteins, and the bacteriophages grown in the presence of P32 had labeled DNA. So the differential labeling thus enables them to identify DNA and not the proteins or the genetic material. Mixing labeled phages. Hershey and Chase mixed the labeled phages with unlabeled E. coli and allowed the bacteriophages to attack and inject their genetic material. Here you can see the sulfur radio-labeled bacteriophage had radioactivity coated on its protein, which is outside the cell, and the phages were allowed to infect the bacteria, and it was found that no radioactivity entered into the cell. Similarly, the DNA of the phages were radio-labeled with phosphorus, and these phages were allowed to infect the bacteria, and they found that the radioactivity entered into the cell. Later, after the infection, before the lysis of the bacteria, the bacterial cells were gently agitated in a blender to cause loosening the adhering phase particles. According to their observations, it was observed that only P32 was found associated with the bacterial cells, and S35 was found in the surrounding medium and not in the bacterial cells. When phage progeny was studied for radioactivity, it was found that P32 was carried inside the cell and not S35. Here you can see the entire procedure of the experiment. Sulfur labeled protein capsule were allowed to infect the bacterial cell. The virion coat remains outside the cell. The DNA or the RNA enters into the cell, and the radioactivity was found outside the cell. On the right side you can see the phosphorus labeled DNA. Infection was done, then blending was done, then centrifugation showed that the phosphorus was found within the cell, indicating that the DNA is the genetic material. What is the conclusion of Hershey-Chase experiment? These results clearly indicate only DNA and not the protein coat entered the bacterial cells. Hershey and Chase thus conclusively proved that it was DNA and not the protein which carries the hereditary information from the virus to the bacteria. Now, we have come to know that DNA is the genetic material and not the protein. Now, what are the major events in the genetic calendar? In 1865, it was found that genes are particulate factors. In 1871, discovery of nucleic acids was done. In 1903, chromosomes were found to be hereditary units. In 1910, genes were found to lie on the chromosomes. In 1913, chromosomes are the linear arrays of genes. In 1927, mutations are physical changes in genes. And in 1931, recombination occurs by crossing over. In 1944, it was found that DNA is the genetic material. In 1945, a gene codes for protein. In 1951, first protein sequence was demonstrated. In 1953, DNA is a double helix. In 1958, DNA replicates semi-conservatively. In 1961, genetic code is triplet. In 1977, eukaryotic genes are interrupted and DNA can be sequenced. In 1995, bacterial genomes were sequenced. In 2001, human genome sequenced. So today we discussed about molecular genetics, part 1, introduction to molecular genetics, gene and DNA as the genetic material. Thank you. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Post your questions in the comment box. We will reply with appropriate answers. Thank you very much.